something that we're going to hold you accountable for at the moment, but I want to take this opportunity because this is probably the one time you're most interested in DNA and how it's copied, to just put a little bit of nugget of information out there. We spent a bunch of time last time in lab building models of DNA using gumdrops and toothpicks, right? And you built them and you said, okay, different layers of gumdrops are either phosphate sugars, A's, C's, C's, and G's, and you put them all together. Now, I want you to look at this DNA double helix up here, and tell me if you see anything different between the two strands of DNA. So, between the two strands of DNA, is there anything different about the shape, the orientation? Just take a peek.
Different than what, each other?
Yeah, so is one strand different than the other in any way?
The bottom.
Not in terms of whether it's a complementary sequence or not. We already have established that. But do you see any other visual differences between the two strands that are up here?
Yeah, the right side.
Top right side?
Sorry, bottom of the right side?
Yeah.
That thing on the other side is there.
That thing on the other side there, it's hard for me to figure out what you want me to point at.
Yeah, it's the OH. It's the fourth one down. There's a loose OH on the right side of the left one.
Right, loose OH on over here.
Yeah, that over there.
Yeah.
Yeah.
So if you look at the actual structure of the sugars and phosphates on each side, one of the things that I want you to notice is that they actually are flipped relative to each other. So the two strands of DNA, they actually don't run parallel to each other like this. They actually run anti-parallel. So they're flipped 180 degrees relative to each other. And that's the way that the hydrogen bonding works between the bases. And that's also important for when the enzyme that can catalyze the formation of the complementary strand, it only proceeds in a particular direction as it makes the copy. Now, I'm only telling you this now because this is the one time in the whole semester where we're really thinking a lot about DNA and its copying. But when we talk about some biotechnology stuff later on, like the polymerase chain reaction, or PCR, remembering back to this will be helpful for actually understanding how the process works. Yes?
Is that ATP down there?
This?
Yes.
It
right
is
there.
ATP, but instead of having an adenine, it has a thymine.
So, okay.
So it's TTP.
Okay, and then that is what, in a similar process, I'm noticing that those two phosphate groups are ejected off.
Yeah.
That molecule floating around is eventually hooked in for a DNA sequencing?
Yeah, well, it's hooked in here because it's complementary to the A over here. And so the energy to actually form this new bond between the TTP and the growing DNA strand is provided from those phosphates.
Okay, and actually my sort of general question really is all the material, I don't know, I feel like this will be obvious once I say it out loud, but all the material for DNA is already floating around inside your cell, and then when that strand is there, that's when they're like, oh, and they start hooking on?
Yeah, so all the material is floating around, and when DNA polymerase and a whole bunch of other enzymes assemble and associate with it, they initiate the process of actually copying it. But yeah, all those materials are available. And they get there by basically diffusing in and happening to be there at the right time at the right place. Okay, so our next step, if you got, I'm going to do something kind of experimental here. I know it's risky, but why not? I want each group to come over here and get one of those strips of paper and a pair of scissors. Some of the scissors are blunt, and so they're safe, and some of them are pointy, and so they're sharp. So please don't hurt yourself.